Thank you very much. A real pleasure to be here in this beautiful part of the world. Always feel very lucky to come back to Australia. This is my second time back here this year, actually. So it's great to be back, and it's great to see so many of you here and lots of really good traction and excitement and discussion around this topic of soils. Such an important resource, of course, that really is the foundation of all of your businesses, that your productivity, your profitability, very much dependent on the function and the health and the status of your soils. So, of course, it is something that we do need more regular boot camps on, and to focus on this important topic and other things that we'll also hear about through the course of today. So let's dive into it, because I'm mindful that I'm up speaking first, and I don't want to go over time and eat into the other great speakers' time. So let's dive straight into it. So when we talk about soil health, this kind of broad topic, this broad term, it's a bit of a subjective term, can be a bit of a fuzzy term, depends on your context, what you see, how you interpret this. And, soil health for what? For pasture, for productivity, for cropping, soil health for nature conservation. It depends. It depends what the context is. But beyond some of those differences, we could often kind of break soil health down into these three components, or at least some of the properties of the soil that we could measure, that we could manage, that we can kind of work towards improving. And so we have this idea, and I think the three-legged stool really captures the essence of this idea really nicely, that the soil has chemical components, physical and biological components, properties, and that they are all important. Just like if one of these legs was a bit loose and rickety, the functionality of that stool is not going to be very, very useful. Similarly, if one of these pieces of the puzzle of your soils, if one of those chemical, physical, or biological properties are also mismanaged or not optimized, of course, the function of your soil will also be compromised. So I think this three-legged stool is really important. It brings balance to this idea that they are all important. And in soil science, we've been, traditionally speaking, very focused on the soil chemistry. We all take soil tests, we look at our pH, we look at our nutrient availability. We've generally been talking about the importance of soil structure and soil physics, but it's been this biology has been the piece that maybe has been somewhat neglected, traditionally speaking. And we're going through that paradigm shift now, where we are bringing new knowledge into this three-legged stool. We're filling in the gaps with the soil biology in terms of understanding how they sit in here and the contribution that they can make to soil health and to soil function. And so it's a very exciting time in the world of soils. We have a lot of emerging ideas, a lot of new concepts out there in terms of what biology can do, the potential, and how they may help us both increase the sustainability of our soil, managing our soil resource, as well as our productivity and profitability. So how do we improve that soil health? Soil biology, a big piece of that puzzle. Generally, I'm going to talk quite a bit about roots today, and roots are a key driver below ground carbon. What happens below ground is really what drives a lot of these changes and improvements in that soil health, in that soil quality. So as we'll talk about, a big piece of the puzzle is about how you are managing your roots. But beyond that, we have these kind of very simplistic principles and ideas for soil management, and that's why these principles, I don't say simplistic as some kind of a criticism there. That's why they are so excellent. Keep it simple. And that's why I think it's so great that the soil health principles are being very much widely discussed all around the world now. They provide these overall umbrella guidelines that really cut through some of the complexity of soil, some of these chemical interactions, some of this vast diversity of all this biology. We have so many knowledge gaps. We actually have very little understanding. We have some regards we have lots of understanding, in some regards we have very little understanding. And that's the beautiful thing about following these macro level guidelines. You know what, despite all of that complexity underneath that soil, if you follow these general guidelines as best as you can within your context, applying these principles, that is an overall strategy that generally pretty much seems to help to improve those soils. So of course, minimizing disturbance, diversity, keeping that soil covered, living roots, and integrating livestock. And so we follow these macro principles, and soils, they can be turned around. They can make a difference in that functionality of those soils. And I think that's why these soil health principles are so excellent, that they are simple, that they provide an easy way to manage this very complex resource. So when we think about soil health and that word health, we typically associate that word health with living things, with life. I'm not so I'm curious about the health of this tin here. I'm not so curious about the health of the rocks or the table, inanimate objects. I'm interested in the health of living things, of my organs, my body, plants, animals, the organisms in the soil. We took that word health and sickness. Health, it very much links to living things, to biology. And so when we think about soil health, it's, well, I've got to take care of the living component of those soils, those living resources, those organisms. And so we would typically think about strategies to feed the soil. When we talk about living things, they all need to eat, they need shelter, they need to eat. So how do we feed that soil in order to feed that soil biology, to drive some of those changes in soil health? So we would do some of these important practices like stubble retention, keeping that soil covered with those litters, maybe good grazing management, not too much overgrazing, maybe leaving some residuals there, trampling, again, a bit of that food in. Again, livestock integration for those croppers in the room. And, other things around composts, manures, organic amendments. These types of inputs are what ultimately, and practices that ultimately help to feed the soil, feed that soil biology, keep that soil healthy from that living point of view. And that's those organisms there that we're trying to increase and improve the health of, is this soil biology. And when I say soil biology, I mean this vast, vast and complex world of micro things like bacteria and fungi, all the way through up to some of the larger things like insects, small insects, larger insects, through to earthworms, etc. It is this vast community of life, a very much entangled web of organisms interacting with each other, eating that carbon that we are bringing in, be that from residues, manures, etc., eating that carbon, digesting that as their food source, growing their bodies. And as those organisms grow and die, they perform all sorts of functions. And those biological functions is what helps plants grow, that helps improve that overall, that soil health. So just some nice visual imagery there of this community of life that exists. Just officially earlier this year, we had a study that came out that has officially declared soils as the most biodiverse ecosystems on the planet. So they really are a rich reserve, particularly amongst the microorganisms, a rich reserve of genes, of genetic information that can be harnessed. Many of our medicines come from microorganisms from the soil, for example. These microorganisms produce all sorts of bioactive compounds and they excrete those into the soil. And these have all sorts of functions in terms of, okay, some are medicines for us, some are also nutrients for plants, stimulants for plants, compounds that can suppress pathogens, all sorts of things, different things that they do. Vast array of these various compounds that exists. And if we think about this piece of this puzzle of soil health and soil biology, if we step back from this conversation about soil health for a second and talk about soil formation, the process of soil formation is a biological process, meaning that we have geology, volcanic activity, tectonic upheaval. Geological material comes to the surface and we begin the process of turning that rock into soil. And that is partly driven by some weathering, rain and cracking and other things, other non-biological forces there. But mainly the way in which we form soils is by having living things, biology, grow on that rock and over time turn that rock into soil. And that starts with things like mosses and algae and lichens, bacteria, micro things that grow on those rocks and begin to extract nutrients out of the rock and incorporate it into their bodies. And when they then die, they grow and die over many, many thousands of years, over many successive generations. When they die, of course, they are depositing their dead bodies. And those dead bodies is made up of carbon. So we are beginning to mix carbon in with the rock. And as we mix carbon and rock together, This is how we form soil. That's what soil is. So there is no soil without biology. Biology with soil without biology is just geology. And this is that point. We have geology on the moon, we have geology on Mars, we have rocks there, but we don't have life, so we don't have soil. So biology is the process that drives soil formation as these things grow on there, and then later on higher plants can grow and they begin to grow and die. Now they're depositing more carbon into that rock. Annuals, pioneer species, they begin that process. And then we move on to some of our perennials and onwards into trees. And all along this process, we are increasing topsoil depth. We are forming soil through the biological interactions with that rock. So in that context of soil formation and biology being key to soil formation, it is also the same in soil health discussion. So if we're trying to improve our soil health, well then biology is a key tool that will drive changes in soil health in exactly the same way that it is responsible for driving processes of soil formation. So biology, they are a very important piece that we are now filling those gaps in. And that biology in the soil can do all sorts of other things in terms of changing that soil, such as changing the soil physics, the structure of the soil. And when some of those biological organisms, particularly the microorganisms, they release some of these compounds into the environment, which are very sticky, gluey substances that help to start to stick soil particles together, forming aggregates. And when we stick particles together, forming aggregates, by default, we create pore space in between those aggregates. And that pore space is what helps that soil to breathe. So this is how, of course, we get water to infiltrate, as we can see on this image, but it is also that porosity in which we get oxygen down into the soil so that those organisms, the life in the soil, can breathe. And they can breathe in that oxygen and release carbon dioxide. And so that carbon dioxide in a well-structured soil can also escape out of that soil and come out of that porosity so that the plants can breathe back in that carbon dioxide and keep that cycle going. So yes, biology, and then of course earthworms, they will channel and tunnel. They will also improve the soil physical condition. So biology impacts soil physics. They are interconnected. And biology also impacts soil chemistry and the nutrient availability and the nutrient cycling in our soils as well. And we're going to spend a few minutes here now talking about how biology drives nutrient cycling. And this is, again, some of the new ideas and the emerging paradigm around biological processes that help to make nutrients available. It's not just about the soluble nutrients that are soluble in the soil water that are available. This is this classic idea of soil, when we've been very stuck in that soil chemistry paradigm, we say that, well, the nutrients need to be soluble to be available. And the plants will drink water from the soil, take up that water, and with that water, they will take up nutrients. Now, this is true. It's not that that isn't factual, but it's just that it is a piece of the puzzle. It is part of the story. There are other ways in which plants can also acquire nutrients. So the funny thing about soluble nutrients, and this is why many of our fertilizers that we use are water soluble, because we've been in that paradigm saying, well, we have to apply these water-soluble nutrients. We apply fertilizers, they will quickly solubilize, therefore they will quickly become available, and the plant can take those up from that soluble pool. But that is a double-edged sword, because yes, those water-soluble nutrients do become very available very quickly, and yes, the plants can take those up. But those highly soluble nutrients are also highly prone to losses. They're also prone to locking up in the soil. They're prone to leaching or off-gassing in the context of nitrogen. So we've also been very inefficient with these soluble fertilizers because one half of them gets very well taken up, but the other half is very much lost from the system or locked up and becomes unavailable. So the point then is we have soil tests that we've used over many last decades where we've pretty much been measuring the nutrients in the soil pretty much here at this soluble fractions, maybe some of these exchangeable or medium-term supply. A typical soil test will measure just these. But you do have more nutrients in your soils, and that's what is called the total, and the total pool. And these are nutrients that are present in the soils, but not necessarily, in the traditional definition, plant available, because they're not soluble. They are there, but they're locked up and unavailable. And the key to making those available is through biology, is through biological interactions. The organisms in the soils can take these locked up nutrients, unlock them, and make them available. And so when we do a soil test, we should be also asking the lab to give us a picture of the total pool, because this tells you really what your total nutrient supply is. And we will find, I can give you some examples, your total pool here, it depends on different soil types, of course. So a sandier soil is going to have a much smaller total pool. A big heavy clay soil is going to have a much larger pool. So of course it's going to depend on different soil types. But just to give you some guidelines, some of those total pools of nutrients can easily be 10 times more or more than that, than what we would typically measure in the available pool. So the essence of this is that soils do have, and again, maybe not light sand, not very shallow soils and very light sandy soils, but many soils do have actually quite ample amounts of nutrients in them. Really, the question is, how do we unlock and cycle those? And that's where we need some of those biological interactions to support that process. So yes, plants can unlock some of those nutrients. Plants can do this themselves, and they do that through some of those root exudates, whereby they will release organic acids, they will release hydrogen, they'll acidify the soil to strip some of those nutrients out. They can release special enzymes that can also solubilize and pull nutrients off the soil. So yes, plants can do it themselves with releasing some of these root exudates like organic acids, hydrogen. This is an example of phosphorus. So a special enzyme there they release called phosphatases. And this is a compound that's very good at grabbing phosphorus and making that available. But plants can also form partnership with the microorganisms. They can also feed the fungi, particularly mycorrhizal fungi. And those fungi are very, very good at unlocking phosphorus and making that available. Plants can also feed bacteria. And those bacteria, many bacteria, are also very good phosphorus solubilizers, or generally other nutrient solubilizers. And the point here is that yes, plants can get nutrients out of the soil themselves, but the microorganisms can do it much, much better. And that is because the microorganisms in the soil, those bacteria and fungi, they exist in far, far greater diversity. It's that genetic diversity that I mentioned. And with that vast diversity of organisms, that vast diversity of life, comes a vast diversity of strategies, of mechanisms, of tools in the toolbox in which they can use and draw from to unlock and access and cycle nutrients. So the point is here that microbes can do this much, much better than the plant can. And that is why then the plant will invest in those organisms and send photosynthetic sugars down to feed them in order to support that nutrient cycling process. So, and we also have now part of this new paradigm. Our previous soil chemistry dominated paradigm was very much saying that plants take up inorganic nutrients, simple ions, just simple small compounds is what the plant takes up. Here's an example of nitrogen, things like nitrate or ammonium. Type in the nitrogen cycle into Google Images and you'll come up with hundreds and hundreds of images of the global nitrogen cycle, how nitrogen moves from the atmosphere back into the soils, around waters, through rivers, etc., in and out of plants and animals, etc. And in every single one of those images, it will demonstrate that the two forms of nitrogen that enter the plant are ammonium and nitrate. And it is not that that is incorrect, it's just that it is not the full story. Plants can take up larger, more complex molecules like organic forms of nitrogen, such as amino acids, little chains of amino acids called peptides, or even larger chains of these called proteins. These are, relatively speaking, larger, more complex molecules. And because of that larger, complex size, we thought that plants couldn't take them up. So we had this whole paradigm of, well, plants take up these small, simple things. So, of course, all of our fertilizers that we use are based around these. And again, these are part of the picture. And yes, they are valid. They are highly available. Yes, the plant can take those up. But again, they are also highly prone to the losses. When we talk about nitrate leaching, well, that's happening here. When we talk about nitrous oxides going off to the atmosphere, it comes from here. When we talk about ammonia gas up into the atmosphere, it comes from here. These are also the very reactive, volatile forms that are prone to these losses. So plants actually specifically release particular root exudates that help them access and unlock some of those larger, more complex pools. And these forms of nitrogen are actually very, very favorable, very beneficial for the plant to use, because the plant, they're very metabolically efficient or energetically efficient, meaning when the plant takes up amino acids and proteins, it's already got it in a favorable form that the plant can then just utilize. Whereas when it takes up these inorganic forms, it actually has to expend energy to turn those into amino acids and then turn them into proteins. You all know that when you will want to build protein. We want protein for good grain quality, for feed quality. Plants want proteins. That is what the goal is. They're trying to produce good quality proteins. And these are just the building blocks to those proteins. And so the plant will expend energy to convert those into proteins. But if we provide some of these organic forms, which are already halfway up this pathway, well, then we are saving the plant's energy to finish the job. And when we save the plant's energy, that saved energy can be used for other growth processes, deeper roots, more tillers, filling more grain, more biomass, etc., whatever it may be. And then we also have now, some of you may have come across this idea of the rhizophagy cycle. We even know that plants actually can also absorb and take up through their roots massive things, ginormous things like bacteria. Ginormous things, relatively speaking, to these little things back here, I mean. Even whole bacteria can get engulfed into the root tips. As those root tips are growing through the soil, the cells of the root tips are dividing, and bacteria get engulfed or internalized into those roots, where those bacteria become endophytes, or they live inside the plant, and the plant actually can send special chemicals to those roots to strip out nutrients out of the bacterial cell wall. So the plants are literally eating the bacteria for their nutrition. So these are emerging ideas that plants actually can acquire nutrients in many different means. Other means, it doesn't have to be applied just in these more simplistic forms. And this is just an example of that rhizophagy cycle that I just mentioned there, whereby bacteria will become internalized, they'll be taken up, stripped of nutrients. And what's also interesting about this cycle is that then, once the nutrients have been stripped out of those cell walls, the plant will actually expel the bacteria out of some of those root hairs. And those bacteria get expelled back out into the soil environment, where they rebuild their cell walls, they rebuild their bodies, so they recharge with nutrients. And then they are attracted back to the root tips, because in those root tips we also have lots of root exudates, foods, sugars, and special chemicals that attract those bacteria to go back to the root tips and repeat that cycle. So here we have an example of nutrients that are not in the soluble form, that are not water soluble, but are still available and being able to be taken up and utilized by the plant. So actually we don't want nutrients to be soluble, we want them to be insoluble and available. But that goes against a simple chemical paradigm that says, well, they must be soluble to be available. But actually, we want them to be insoluble and available. And there's a perfect example there. Those nutrients trapped inside those bacterial bodies, those bacteria are a reservoir to hold nutrients in a stable form that is not going to leach or lock up, etc. So they are unavailable in that traditional definition. They are, but they are still actually very available through these biological means. And we have some of these other interesting ideas like quorum sensing. Some of you that listened to Christine Jones would, of course, come across this. And this is just another interesting area whereby when bacteria are in low density, they behave a certain way. So we have individual behaviors. But once they start to multiply and grow into more higher density, as I mentioned, microorganisms, they release all these compounds, bioactive substances, into their environment. And when some of these chemicals, these biochemicals, start to increase to a certain concentration, the concentration of that chemical can unlock genetic expression in the bacteria, meaning it can switch on genes inside the bacteria that are not able to be switched on when those bacteria are at low density, when we don't have enough concentration of these special chemicals that can trigger this process. And why that's important is it just simply means that we have population behaviors versus individual behaviors, and the things that bacteria can do at higher populations, at these higher densities, can be very, very different to what they can do here. So there is potential in our soil biology to unlock their functions, but only when we optimize soil health and improve that soil quality and improve that soil biological population to such an extent where we can begin to observe things that are not observable otherwise. And so I think this is a very exciting, still emerging. I think there's a lot of practical, how do we kind of apply some of this practically, some of these knowledge gaps that we definitely still need to fill. But nonetheless, it illustrates some of the potential of that soil biology that is still very unknown, therefore untapped. Here's a nice example. Some of you might have seen this one from quite a few years ago. This was an interesting study on some maize, a land race. Land race meaning it's not a variety, it's a mixed population. So it's not an identical population like a variety would be. It's a mixture of genetics. So that's kind of called a land race. And this was from central Mexico, where they identified these soils are very, very low in nitrogen, very nutrient poor soils, nitrogen poor soils. And they noticed this unusual habit of these maize populations. They produce these aerial roots that you can see here, these buttress roots, but they were covered in this thick, sugary mucilage that you can see here. And what they determined is that actually these maize plants produce these specialized aerial roots, and in that sugary mucilage there is a whole bunch of chemicals that are attracting nitrogen-fixing bacteria. And it turns out that this maize was fixing its own nitrogen, growing in these nitrogen-depleted soils. And they determined that, and it's quite a wide range here, it's a lot of variability, and again, that's because it's a land race, it's not uniform genetics, so it's mixed genetics in these maize plants. But they demonstrated that these maize plants could fix anywhere from 30 to 80% of their own nitrogen needs, of their own nitrogen requirements. They could fix in symbiotic partnership with free-living associative nitrogen-fixing bacteria, not rhizobium, not legumes, other bacteria that can also do that same function and associate with grasses and cereals, et cetera, and all sorts of other plants. So this is just a nice example of some untapped potential. Now we're looking at trying to breed some of the genetics from these varieties into modern varieties and give modern varieties an ability to fix their own nitrogen, not through genetic engineering, just through general plant breeding. But here's what's also interesting. I was just in Kansas earlier this year, back in January, and a farmer there told me that some farmers growing more modern maize varieties that we would say don't have this ability, he was saying that some farmers who are down the pathway of soil health practices and principles, who are really increasing their soil health and their soil biology, some of these farmers are beginning to see this be expressed in the modern varieties of the plants. Meaning it is not just the genetics of the variety, yes, although that is a big part of it, it's also the environment and the presence of good soil health, good soil biology, and the biology can actually change the plant development, can make the plant express, unlock genes of the plant, so that even some of the modern US varieties, for example, may indeed already have this ability hidden, embedded in their genetics, but we're just not turning it on because of the way we manage the soil, the way we manage the system. And this is just for interest's sake, this is an ornamental plant, but I'm just bouncing off this idea here. We have a more recent study on this ornamental plant, and I just want to point out one thing. It's the same thing. They were studying these aerial roots and this mucilage, again, full of nitrogen-fixing bacteria. In this study, they identified that there was also a fungus in there. There was a fungi growing in that bubble, in that mucilage. And this fungus was producing a broad-spectrum antibiotic that was suppressing most other bacteria apart from the N-fixing bacteria, who was tolerant to that. So there was a partnership there between this bacteria and fungi, where the fungi is suppressing the competitors, enabling this N-fixing bacteria to colonize that root and feed on that mucilage, providing the sugar to then fix atmospheric nitrogen straight out of the air into this plant. So I share that more so, again, for interest's sake, just because there are many fascinating things out there that we are still yet to uncover. And so that then brings us to this point that plants Just like us, we are a partnership with all of the bacteria in our guts, with our gut biome, gut microbiome. Same for plants. And this is kind of coining this term now, holobiont, which we are holobionts, plants are holobionts, meaning plants aren't just plants. Plants are plants plus their associated microbes. We're not just human, we're human plus our associated microbiome. So when we think about studying plants or studying humans, we can't separate these things, which is what we've been doing in reductionist science. We do plant science, we study plants over there, we do microbiology, and we do microbiology over there. But actually, plants and microbes are inseparable, just like we are not. And actually, if we want to study plants and improve the potential of these things, we have to study them as one whole unit of the plant plus its associated microbiome. And this is this kind of idea of more a holistic or holobiome. So we have fascinating things now that we are uncovering, that microbes grow not just on and around and in those roots, but also on and around and inside all of the above ground biomass as well. And this is just a nice visual example of what I was talking about there earlier with the soil health practices, maybe inducing some of these changes in those maize plants. Plant breeding goes microbial. Here's a nice example. You could take the exact same variety of plant and you could grow it in two different contexts with two different soil microbiomes, two different biological balances. And of course, those different microbiomes will grow around that plant. So we have two different microbial balances or composition or different groups of organisms, and they are releasing all of those biostimulants, biocompounds. Some of the plant will detect those and it will induce changes in the genetic function of the plant. So we can change, for example, the phenotype. It's the exact same genetics, exact same varieties, but we could express more growth, for example, like above ground with the leaf, or express more growth below ground on those roots. Even though the genetics of the variety are identical, G by E, genes by environment, the environment in which we grow the plants will influence the outcome. So this is then the importance of improving soil health from this soil biological point of view. So microbes help plants grow, and when they help plants grow, they help plants photosynthesize. Breathe in carbon from the air and use that carbon to grow their bodies. And of course, we need sunlight to provide energy for this. And that carbon gets taken out of the air and fixed into sugar, into glucose, into the product of photosynthesis. And that's how plants grow their biomass. And of course, thankfully for us, they also produce that little bit of oxygen as a byproduct. So we are fixing carbon out of the air and turning that into a form that is usable in which plants can grow. Now, some of that photosynthetic carbon stays above ground and grows shoots and leaves, etc. And some of that carbon is sent to below ground and produces those root mass and that root litter. And of course, the more roots we can grow, the deeper the roots, the more branching those roots, the more different root architectures that we can grow and produce down below, that is going to particularly increase and change the soil physics. Look at all those root channels that have penetrated down into that soil, and as those roots grow and die, they are leaving these channels and tunnels influencing the soil physics. So yes, plants and roots very much drive changes in soil physics as well. And the deeper that we can get those roots over time, the deeper we can increase our soil depth. And the deeper you can grow your topsoils and make your soils help your plants access deeper soil, simply means you're going to access a whole lot more nutrients and moisture deeper down. And that's what's going to add resilience to your system. Now, it is also those roots and that below ground carbon that helps to build soil organic matter, to build soil carbon. And actually, we have lots of studies that show that actually it is roots and not shoots that make soil organic matter, that build soil organic matter. What happens above ground is interconnected. We don't have roots without shoots, of course, but it is more direct, the action of roots that build soil organic matter. What happens above ground is not the main driver of soil carbon formation. Now, above ground biomass is still important. That's your yield. That's what you're going to be harvesting. Now, some of that surface shoots and litter, again, that can help to provide other important soil functions in terms of soil protection, moisture conservation, habitat for insects, food for earthworms, rain protection, wind protection. So yes, all of that shoot material is very important on the surface, but it doesn't really contribute very much to building that soil organic matter. So we've got to think about more below ground carbon if we want to increase soil carbon. And what about those root exudates? I mentioned those photosynthetic products, sugars and carbohydrates and other compounds that are the products of photosynthesis that get sent down to the roots, and actually not just to grow root mass, but they are excreted or leaked out of those roots. The liquid carbon pathway, as Christine would also call that. What about these root exudates? What impact do they play on building soil carbon? And it turns out that they also play a very, very important role. And this was a study here that was looking at this. And they identified that those root exudates are anywhere from 2 to 13 times more efficient at building soil organic matter than the root litter itself. So the root litter is about 5 times more efficient than the shoot litter. So you'd need about five times less, an equivalent amount of biomass of roots versus shoots to build soil organic matter. But it turns out that these root exudates are even more efficient still, 2 to 13 times more effective. And there's a few factors here, but that really comes down to something quite simple, which is called the carbon use efficiency. And all of you livestock people in the room will understand this very simply. We're just talking about the digestibility of the carbon. Here we have high digestible carbon. Here we have lower digestible carbon. Now this over time, yes, it will break down, it will decompose, and it will slowly help to build soil organic matter. But that long amount of time, that slow decay, a slow decomposition that happens here, that slow process means that there's more opportunities for losses, for oxidation, for CO2 to be lost. The high digestibility of this carbon means that microorganisms crowding around those roots will immediately and rapidly consume that carbon and grow their bodies. They're not spending a long time to digest, break down. We've got to cut all that complex carbon up and cut it up and get it smaller and smaller, digest it, until it's small enough, until it's high digestibility that they can then absorb it and grow. We don't need to wait for all of that process, and it's not to detract from that. Again, it plays its role, soil protection, slow pathway. But here we have the fast pathway whereby this carbon is high digestible. Microbes will immediately eat this and start growing and feeding and reproducing and performing function. And that's why this is a more efficient pathway because of this high digestibility. And the efficiency by which we grow microbial biomass is important because we now know that soil organic matter itself, soil organic carbon, is made up of around about 50-odd% dead microbial bodies. This is called microbial necromass. And this necromass is when those microbes grow. First they grow biomass, so they reproduce and they are growing biomass while they are living. And when they die, they form this necromass, dead bodies. And it is this necromass that actually makes up around about 50-odd% of what is soil carbon. Now, we used to previously think that soil organic matter and soil carbon was all made up of plants that are slowly decaying, decay, and at the end point of decomposition, that plant material turns into humus or into soil organic matter. And yes, again, it's not that that is false, it's just that it's half the story. Because at some point in that decay, microbes start to eat it and ingest it. And when a microbe ingests that carbon, that is no longer a plant. That carbon becomes microbial. It is now in the body of that organism. So it is the microbial contribution or the microbial pathway. And when those microbes eat that plant material and they again grow and die and they form that necromass, it is that necromass that is a particularly stable fraction of carbon in the soil when it sticks to the soil particles and gets kind of glued and chemically stuck. So it forms a very stable fraction of your soil organic carbon. And as the study suggests here, it's around about 50-odd%. So if we want to optimize both pathways to build soil carbon, the plant contribution and the microbial contribution, we simply need to embrace those wonderful soil health principles of keep soil armor, keep the soil covered, and living roots. Green cover, brown cover. We want both of them. And this is the importance of the green cover or the living roots principle. The soil health principle of living roots was way ahead of the science. Now the science has realized, oh, actually dead microbial mass is a big part of soil organic matter. The best way to facilitate that pathway is living roots. And those living root principle was on point. So we also have a great way to stimulate below ground carbon and some of this rooting and root exudate process, and that's with, dare I say it, livestock. Livestock are a wonderful tool to enhance and improve and increase this process. Here we have a great study on a light sandy soil. It's from the US, so you can take it with a bit of a grain of salt, but it illustrates an important principle here, where they took a pasture, they split it in half. On one half they cut and mowed, on the other half they introduced livestock and they grazed. So we have grazing exclusion with cutting and mowing, and grazing inclusion with the animal being present. And what they found here is that where they excluded those animals, grazing exclusion was associated with less below ground carbon allocation. So that's going to mean less root biomass. They also measured less root exudation, and therefore they also measured less microbial biomass, and all of that resulted in lower soil organic carbon. So the interaction of the animal is very positive in encouraging rooting responses, below ground carbon, root exudation, microbial biomass, more soil carbon formation. Here's another one on this topic of livestock exclusion from the UK. So fine, take it with a grain of salt, but again, principle is very important here. Cessation of grazing causes biodiversity loss. So we show that cessation of grazing leads to strongly decreased diversity of most groups of soil microbes and soil fauna, some of the insects, particularly some of the rare taxa. Overall, our results indicate that the exclusion of domesticated herbivores from historically grazed montane grasslands, kind of hillside, mountainous grasslands, has far-ranging negative consequences for diversity of the below-ground food webs, and this underscores the importance of grazers for maintaining the diversity of below-ground communities, which play a central role in ecosystem functioning. So here we have another nice example illustrating that grazing exclusion is associated with loss of biodiversity of some of those microorganisms and soil insects. And what about another one? Here, a nice example of grazing exclusion with a little bit of leaving litters and residuals on the surface without some trampling, versus where we have animal inclusion and we get a little bit of that animal impact, that interaction of small bit of disturbance, or there's another dirty word, disturbance, or we're not supposed to disturb the soil. But here we have a little bit of mild disturbance from the animal impact, a little bit of integration of those surface litters into that soil. And by that mild disturbance, surface disturbance, integrating that litter, what do? Very simply, we increased the accessibility of that carbon, of that litter, to the microbes. We've got a lot of hungry mouths down here, and they're microorganisms. They're not very mobile in the soil. They're just where they are. So if we don't integrate, you've got a lot of hungry mouths down here that can't get to that surface litter. But with a little bit of integration, now we have increased accessibility of that carbon to those microbes so that they can decompose, so that they can grow their bodies, so that they can grow and die and form that necromass and build soil organic carbon. And that's exactly what they found, is that by integrating, we actually stimulated microbial activity, and that actually led to greater carbon dioxide losses by about 12%. We increased CO2 losses. So if we were measuring CO2 as an indicator of soil biological activity, we might alone, if we were just measuring that, we would say, oh, this was not good, because we increased the losses through CO2. But although we did increase the losses, we also increased the gains. And actually these necromass components building the soil organic matter, we also increased soil carbon. You can't quite see it here, but it's by 76% that we increased the soil carbon. So we increased the losses and we increased the gains because we stimulated the cycle. Okay, it's not that losing carbon dioxide out the top is inherently a bad thing. It's part of the cycle. We want some to come up, some to be breathed back in, and some to cycled. Now, that sounds a bit of a paradox. How do we increase losses and increase gains? Of course, this is just dependent on time. So here we just have that carbon all still locked up. It's just locked up and not yet functional. So eventually it's going to decay and do whatever it will do. But in the period of time of the study, we stimulated carbon cycling. Yeah, we increased some losses, but we increased the gains, and those gains more than offset those losses. Now, what about plant species diversity? That also helps to increase soil carbon. So using multi-species pastures is definitely a better strategy for soil carbon. I know this is a very busy slide. I'll just draw your eye to all of the red. So we're comparing multi-species pastures. We're comparing 1, 2 species, 4 species, 8 species, and 16 species. So 16 is the more diverse treatments here. That's the red. And you can see consistently that the red treatments are all higher. We're measuring root carbon, we're measuring soil carbon at the surface, soil carbon at depth, and we're measuring this across time. And you can see consistently that the 16 species plots are consistently more root carbon, more soil carbon, and more carbon over time. But of course, that depends on how we manage those pastures, and rest and recovery is absolutely key to this. The way in which we get those deep roots to access subsoil moisture and access those nutrients at depth is through a nice long rest and recovery period. Full expression above ground will give us full expression below ground. And this is how we restructure soils. This is how roots perform that function. They're not going to do it if we graze too soon to the fully potential. They're not going to do it if we keep the pastures low and we're set stocking. We want long rest and recovery. Full expression above ground will give us full expression below ground. That is how you're going to unlock soil depth and access of those soil resources towards production. So grazing management is key, and diversity really helps that discussion, as I just mentioned, improving soil carbon, but it improves all sorts of other things in terms of course, again, moisture and nutrient access at depth. We have fibrous branching roots, we have tap roots, we have a range of different root architectures that are all doing a slightly different way, slightly different thing to improve that soil. We have different plant species, they're all releasing different root exudates, and those root exudates, again, stimulating the biology over and above what a monoculture could do. So that's the benefit, and of course, in a grazing context and a cover crop context, it's really easy to go highly diverse. A little bit harder in a cropping context, but nonetheless, I'm not going to talk about it today, but nonetheless, even a companion crop or an intercrop, just going from 1 to 2. I've got many other studies and examples that I often talk about. There are big changes in soil chemical, physical, and biological properties when we just go to 2 root systems versus 1. So even that jump from 1 to 2, there are lots of benefits and lots of gains. So, what plant species do we use? Well, it depends on what is your goal. What are you trying to achieve? Do you want N fixation? We might want legumes. Do you want phosphorus release? Buckwheat. Legumes also are good. Brassica is generally quite good at this. Do we want to capture nutrients, prevent them from being lost and leached away? So then we want some deep-rooted plants to capture those subsoil nutrients before they are lost from the system. Nutrient cycling, so we just want to be feeding the biology, of course, to drive some of that. Some plants are very good at suppressing disease. Again, the Brassica family would be a good example there. Some plants are good for producing floral resources, so pollinators. Do we want to attract predators? Do we want to attract beneficials? Then we need some flowering plants. Are we trying to suppress weeds, or maybe a quick-growing plant to shade and outcompete? Do we want to increase soil organic matter or alleviate compaction? Well, then we want deep roots. We want lots of roots and deep roots. So depending on what your goal is, that might influence what plant species you choose. And generally, I think most farmers pretty much want most of these goals. So that means if you want multiple goals, then you're going to need multiple species to achieve those multiple goals. And that's the benefit of those diverse swards. Another quick example here. This is a really neat study, just a lab study, but again, illustrates a really nice point. We're comparing a monoculture of a mustard here with a 4-way mix and a 12-way mix. And what we're looking at is CO2 absorption. So these plants are growing just in a gas chamber where we can measure CO2 depletion. And of course, the more CO2 that's getting depleted out of the atmosphere means that the more that's coming into the plant through photosynthesis. So they found that the CO2 uptake was 2 times higher in that 4-way mix and 3 times higher in that 12-way mix. So with that diversity of plants, diversity of leaves and above ground canopy and architecture, they are capturing sunlight, capturing that important resource more effectively, enabling them to breathe more carbon in, fix more carbon, send that carbon down to the root system, where it also stimulated more microbial activity. So microbial biomass, 8% higher in that 4-way mix, 18% higher in that 12-way mix. as compared to the monoculture. So diversity helps us get more carbon out of the air in the first place. And once we do that, that's half the job, most of the job done. Because then that carbon will be sent down to the soil, down to the roots, where it will grow root matter or produce those root exudates and feed biology and grow biology. All of that will grow and die and turn over and form soil carbon. So this is a big part of what diversity brings, is more CO2 capture. When we use diversity and put some legumes in the mix, of course, they are going to fix that nitrogen for us, and they are going to help fulfill that function, helping us potentially, of course, use less nitrogen fertilizer. And this is a really neat study that was comparing a perennial ryegrass monoculture at a range of different nitrogen fertilizer rates with a couple of different mixes. So with that perennial ryegrass, we just added some timothy and 2 red and white clovers. And to mix 2 here, we also additionally added some plantain and chicory. And across that nitrogen gradient, here's our baseline. You can see at 0 kgs in a monoculture ryegrass, here's the production that they achieved, 6 tons of dry matter. And if we look over here, there's our baseline with 0 fertilizer, monoculture. Over here, 0 fertilizer with some legumes. Well, of course, they overcome the nitrogen issue. Biomass went up. Same with mix 2. So let's go to 120 kgs of nitrogen. Now, of course, we get a big response from that nitrogen under a mono ryegrass, and we've gone up here to just below 10. So let's come over to here on our mix with 0. We've still out-yielded. At 0, we've still out-yielded. Let's go to 240 kgs of nitrogen under that ryegrass. Now here's our target. If we come across here, okay, now we've got this mix is a little lower. It's not much, but it's a little lower. And this mix is here, as you can see, is also on par. So here I'm just illustrating that 240 kgs of nitrogen, we can achieve the same biomass production with 0 kgs of nitrogen just by adding in some diversity. Obviously, particularly some legumes are important there. Here's another nice example of a legume grass mixture. I'm not going to read you out this, I'll just read you the summary points here. Grass legume mixtures can yield more nitrogen than a legume pure stand due to mutual stimulation of nitrogen uptake from both symbiotic and non-symbiotic sources. So we describe how legume proportion modifies the nitrogen acquisition from different sources. What they found is that the symbiotic nitrogen fixation from the legumes was stimulated in those mixtures as compared to a monoculture. Legumes will fix more nitrogen when they are partnered up with a nitrogen-hungry plant like a grass or a cereal, for example, where those plants will be constantly depleting and scavenging nitrogen from the soil, which forces the legume to keep on fixing more nitrogen. So legumes will fix more nitrogen in a partnership than they will under a monoculture. They also found that the uptake of the soil nitrogen from the soil nitrogen pools was also stimulated in the mixtures. And again, that's because we have a nitrogen-hungry plant, fibrous root system. It's going to utilize and access the soil nitrogen more effectively. And they found that overall, the acquired nitrogen, be that from the soil or from infixation, was then used by the mixtures to produce more biomass. So they were more efficient at converting nitrogen into biomass production. Now, nitrogen fertilizers have a lot of negatives. They're a tool. I'm not anti-nitrogen fertilizers, just the dose makes the poison. A little bit is okay. It's too much that then becomes the problem. And here we have a nice example on a grassland from Rothamsted in the UK. Here we have a grassland pasture with a control, no fertilizer. And look at the plant species diversity we have here. Lots of diversity of plants. As soon as we have a 150-year history of fertilization, what happens to that plant species diversity? We lose plants. We begin to have the grasses dominate, take over, and we lose some of that diversity. So there are negatives in terms of biodiversity loss associated with nitrogen enrichment. And equally, like you all know, nitrogen application will suppress rhizobium in legumes. And those other free-living nitrogen-fixing bacteria that I mentioned, like the ones that associate with the maize or others that associate with grasses, they're called associative N-fixing bacteria. They will associate with any plant. They'll just crowd around that root system and perform that same function, not in nodules, but just around that root system. These free-living associative N-fixing bacteria are also suppressed by nitrogen fertilization. So the very process that we want to encourage and support, like N fixation to get free air, N out of the air, and biodiversity to perform those multi-functions, both of these two things are compromised by soil nitrogen applications. So in closing, it's about priming the above ground processes. We want to prime plant growth, photosynthesis, and we can do that with better plant nutrition. We can do that with more plant species diversity, and we can do that more with better grazing management. Longer rest and recoveries is how we're going to prime the above ground. And then we want to, of course, support the below ground cycles of microbial growth, microbial digestion, microbial death and decay, and that's what's going to help to build soil organic matter. And of course, those microbes, as we talked about, are going to help us restructure the soil, improve the physics, cycle the nutrients, make fertility available so that we can reduce our dependency on inputs.